In this lecture, we're going to take a tour of the cell. The cell is going to be the smallest unit of life, and we are going to explore the different parts of the cell, including all of the organelles that are going to function within it. When we categorize living things, we're going to put them into two categories. They can be single-celled or they can be multicellular. Here in these images, I have examples of unicellular and multicellular. So down here on the bottom, we're going to have these unicellular, so they're going to be a single cell. They sometimes will live together looking like they're multicellular, but each cell is going to be by itself. We can then look at something up here, and these are baby squids. They're very, very young squids, and each of these little dots represents a different type of cell. Okay, and so they look pretty crazy. I love this picture. But each of these is going to be made out of thousands and thousands of cells. Okay. Single-celled organisms are going to be called prokaryotes in some situations, and there's going to be a group called the protists. And multicellular organisms are going to be those that we find in the plants, the animals, and most of the fungi. We're going to now talk about the discovery of the cell. So if we go back far enough, people didn't know that everything was made out of smaller units called cells because they're too small to see. And it wasn't until the actual technology was there to kind of enhance our ability to see things very, very small that we were able to discover that things are made out of cells. So the first cell we think was discovered by Robert Hooke in 1665, and he was looking at cork, which is from a tree, and he looked at it under a very crude microscope. So he made something like a microscope, nothing like what we have today, but that would be able to increase the magnification slightly so he could see a little bit more of the fine detail of the cork under the slide. And what he saw were that there are these tiny little holes. And you can see this image of the cork under the microscope. You can see the little tiny holes that are represented in black. And he described those as tiny rooms or cellulae. And you can see that this is where the word cell came from. He called these small sections cells, and he also discovered free-living and parasitic microscopic protists. He looked at sperm cells, blood cells, and much more. So he was able to produce these images that he created, and also wrote a book about minute bodies made by magnifying glasses. So he observed lots of different things under the microscope that no one was able to see prior to the invention of a microscope, which you can see over here on this diagram. So after Robert Hooke, and there was other individuals that started to look at things on a microscopic level, it wasn't until the 19th century, which is the 1800s, that what we call the cell theory was developed. And this is a theory that is saying that all animals and plants are aggregates or just a lot of cells put together. And so you take a human or a plant and you go down to the smallest living unit, it's going to be a cell. Now there are two main scientists that are credited with coming up with the cell theory. There is a German botanist, Matthias Schilden, who proposed that all plants are made out of living cells, and this image in black are different cells that he drew from microscopic viewings. And there was a zoologist studies animals, and his name was Theodor Swan, and he extended the idea to animals. So both Matthias and Theodor came up with these ideas in 1838 and 1839. A zoologist who deals with animals, and he also had all of these drawings of animal cells, and a botanist who studies plants, did it in plants. This is going to lead to a more formal theory called the cell theory. Schledelin and Schwann in 1838 formalized what we call the cell theory. It has three major statements. The first is that all living things, all living organisms are made of cells. The second is that the cell is the fundamental unit of structure, function, and organization of life. And number 3, all cells come from pre-existing cells. That was added a little bit later by another man named Rudolf Virchow. Now, we have changed and modified the cell theory as we've gotten more and more information. And now the modern interpretation of the cell theory is that cells contain the hereditary material and cells process energy. So here we have 2 different cells that we can see they're kind of very brightly colored. They don't normally look like this, but I like pictures that have a lot of color, so these have been added with fluorescence. But you can see the cells right here, and the cell is going to be the fundamental unit of life. Okay, so biologists have since recognized that only cells can produce other cells. You do not get a cell out of nothing. At least at this point in the history of life. We will talk about how we think the first cells ever on the planet were formed, and that takes place in a very different environment that we'll mention in a few lectures. So cells are going to be that indivisible unit, very much like the atom was for the element. If you break down a cell, it is no longer going to be functional for life. Okay, you can break it down and you can get all these different molecules, specifically things like macromolecules that we've talked about, but that is not a living entity. And so the cell is going to be that fundamental unit. This whole theory has then initiated a whole field called cell biology. So people that are cell biologists study cells and how they function, how they work, how they communicate, all these different aspects of the cell. So let's talk a little bit more about what cells are. So cells are going to be made out of a collection of atoms and molecules. So all those things that we talked about in the chemistry lecture and also the molecules, the biological molecules, that's what cells are made out of. So if you go down to the smallest unit of a cell, you're going to get to an atom. Okay? In order for something to be considered living, it's going to have to do a couple things. It's going to have to reproduce, it's going to have to take in matter, and it's going to have to use energy to stay alive. And life is going to result from the collective behavior of all of these different atoms organized into a system. And we've talked about this previously, if you remembered back to the very first lecture, where we talked about this emergent property. So if you take atoms and you arrange them in a very specific way, they can have this emergent property, which is life. So I want to put the cell size in perspective. Okay, so if we look at this image, we are going to start off with your finger, and from your finger we are going to zoom in on kind of the very kind of small corner of your finger. We are going to zoom in, and this is about 2 millimeters, so this is still in the range of something that we can potentially see. Once we go down, we're going to zoom in closer onto these cells that are right beneath the skin, and we can now see some cells that are in here. Okay, this is 0.2 millimeters, so we're now at the point where we would not be able to see this with our eye. If we zoom in on the cell, we can then get to this stage. This is 20 micrometers, which is very small, so you would not be able to see this unless you had a microscope. We can then zoom even further in, and this is going to be a mitochondria, which is an organelle within the cell, so it's going to be smaller than a cellular level. We can then zoom in even further, and we're going to get to a protein level. So this is going to be a protein. Specifically, this is a ribosome that is going to help make proteins. So it's a protein that makes proteins. And if we go even further in on here, we can see this is the protein structure. So this has had all of its conformational folding, and it has a very unique shape. But if you zoom in even closer, you're going to get down to the level of something like an amino acid. An amino acid is just going to be collections of carbons, hydrogens, nitrogens, and oxygens. And if we zoom in even further, we are now going to be at the level of the atom. Okay, so here is going to be, I'm not sure exactly what colors are representing, but this could potentially be oxygen. So this would be an oxygen atom, this could be a nitrogen atom, maybe this is a carbon. So we are going, we started off at the smallest unit, which is the atom. And we're now going to move into kind of this area right here where we're going to start talking about the cell and the different organelles that are inside the cell. Okay, so we've kind of gone through all of these steps. Last time we talked about biological molecules, and now we're going to see what happens when you have biological molecules start to come together and form even larger structures. So cells are going to be the smallest unit of life, but how small can they actually get? Well, this is the smallest cell that has been discovered to date, and this is a tiny bacterial cell, and you could fit 150,000 of these on the very tip of a human hair. So they are absolutely tiny. They don't think that a cell could get much smaller than this because there's not enough room to contain all the materials that life is needed. So there's enormous variety of different sizes of cells that you can find in nature, and they're going to come in all different shapes and have all these different types of functions. If you go to this link, it's going to take you to a size comparison that will show you the different size of different, in this case, I think most of them are viruses and bacteria. Here is a scale, and it's a metric system, so make sure that you are knowing your metric system. But we are going from 1 millimeter to 100 picometers, which this is tiny at the atomic level. But you can see in between kind of the different sizes of, for example, eukaryotic cells, so these are going to be like animals and plant cells, and something like a bacteria cell. So cells are going to come in a variety of different shapes. For example, bacteria are going to come in 3 varieties. They're either spherical, which is a circle, a rod, which kind of looks like a hot dog, and a spiral. Plant cells are often going to be rectangular, and in animals, the cells are going to their shape is going to depend on where the location of the cell is and also its function. So muscle cells, for example, are going to be extremely long, and you can see that in this image. So this is, you're not even seeing a very large portion of the muscle cell. We're going to have nerve cells that are highly branched that we can see in this image. So we would have, it's very hard to tell one cell from another, but kind of this dark middle spot would be a cell and then have branches off of it. And then sperm cells are going to have these long flagellums, which are going to be tails that allow them to swim. The differences in the shapes of the cells are going to reflect differences in their function. So the reason why we have really elongated cells for muscles is that they'll be able to exert force when they contract. And the reason why nerve cells have so many branches is so they can connect to many other cells and transmit kind of this neural information. Now, all cells are going to have to fulfill kind of a set of functions. They're going to have to constantly be intaking raw material. They're going to have to be making energy for them to be able to continue to live, and they're also going to have to reproduce. So this just shows a depiction of all the different type of chemical reactions that are actually going to be happening within the cell. And it's absolutely essential that this is constantly happening. If there ever is a time where a cell is no longer kind of doing these processes, it is going to be dead. Now, there are lots of different types of cells, and they're going to have different strategies. So living things are going to be able to satisfy their needs in different ways. So some cells, which we're going to mainly focus on, things like bacteria, are going to operate as individual cells. So they're all by themselves. They are single units. They do not live together and function together. And they are going to reproduce at really fast rates, and that's going to allow them to survive. Now, other organisms, for example, us, are going to be multicellular. And in the multicellular groups, we're going to have plants, animals, funguses. These are going to have cells, but they're going to work collectively. I found this really cool image. You're going to notice we have the squid, but this is showing larvae of a limpet, a veld chameleon, a dwarf cuttlefish, a mouse embryo, and an amphipod. So all of these are going to be what we call multicellular organisms. Okay, so these life forms are going to be complex. They're going to have thousands upon thousands and sometimes millions and trillions of cells. All of them are going to have different types of cells that are going to function in different ways that allow for kind of a more complex lifestyle. And all the cells are going to be interdependent on each other. So unlike something that's single-celled, multicellular organisms, you can't just take a single cell from them and usually it will not be able to live on its own. So these cells work together and are able to create something larger, this multicellular organism. Now, when we look at all of the cells on Earth, they're going to be able to go into two major categories. The first category is going to be called prokaryotic cells, and these are going to include things like bacteria and another group called archaea. All of these are going to be single-celled organisms. The second group are called eukaryotic cells, or eukaryotes. These are going to include plants, funguses, and animals, and these are going to be mostly multicellular. There are some eukaryotic cells that are single-celled, but they're going to have kind of very different structures and they're going to have different types of organization. Now, if you see here this image, this is going to be a prokaryotic cell and this is going to be a eukaryotic cell. These are not shown to size. Prokaryotes are often very small, but you can just kind of see the difference between these. And we'll talk about what those big differences are. All of the cells on Earth are going to have some of the same basic features, but then there are going to be major differences between these two groups. All cells are going to have what we call a plasma membrane, and this is going to be the membrane that's going to protect the inside of the cell from the outside of the cell. So it is a protective barrier that is going to regulate what can go in and out of the cell. And both prokaryotes and eukaryotes are going to have this. All cells are going to have DNA and they're going to have ribosomes. Now we haven't really talked about ribosomes yet, but we have talked about DNA. So DNA is going to be that heritable material that is going to have all the instructions for proteins and the different things in the bodies or the organisms. Ribosomes are actually the structures that are going to build proteins, and we will talk about ribosomes in depth. But we're going to have both DNA and ribosomes in every single cell. They're absolutely essential for them to be there. So let's do some comparison of prokaryotes and eukaryotes. And we actually have two different eukaryotes on here. We have a plant cell and we have an animal cell, and there are going to be some differences between the plant and the animal. Both the plant cell and animal cell are considered eukaryotes. Now I want to mention what this name means. The name eukaryote means true, so EU means true, and this karyote has to do with something called the karyon or a kernel, and this is referring to what we call the nucleus. So inside of eukaryotes, there is a nucleus that has a membrane around all the DNA. So both the plant and the animal will have this nucleus. So that is what the eukaryote is telling you. It's saying this has a nucleus. Prokaryotes, like the bacterial cell, do not have a nucleus. They're pro, which means before, and this part, karyon, again, is going to be referring to the nucleus. So prokaryotes are organisms that they have no nucleus and they actually evolved prior to the eukaryotes. Now we're going to start going into the depth of all these different little structures inside of the cells. These are called organelles, and you can kind of think of them as little organs like our bodies. We have a spleen, we have a liver, we have lungs and heart. Those are organs, and so organelles are going to be similar in that they're these little structures. They're going to have very specific functions that sustain the life of the cells. Now, all of these are going to have some characteristics that are the same, so I want to just mention those. You're going to see that the DNA is labeled in all three of these, so here's the DNA in purple, We have DNA here in purple and here in purple. This darker spot in the middle is, it's called the nucleus, and we'll talk about it, but the DNA is also just in that area. The next thing that they all have is going to be cytoplasm, which I haven't mentioned before. Cyto means cell and plasm means liquid. So the inside is going to be filled with a liquid called cytoplasm, and all three of these are going to have that. I did mention previously the plasma membrane, so each of them is going to have this barrier that protects it from the external world. And they're also going to have ribosomes that I mentioned down here. You can see them, they're these little tiny dots on this structure, and they're also just floating in the cytoplasm. You can see some of them in here. They might be these little green structures. Now, prokaryotes and eukaryotes are now going to have some very big differences. Now, the first thing to know about prokaryotes is that they are a lot older than the eukaryotic cells. So prokaryotes are going to be the first life forms that show up on the planet about 3.5 billion years ago. Eukaryotes aren't going to show up until about 2.1 billion years ago. And so there's a substantial amount of time, we're talking billions of years, that are kind of when the prokaryotes are all by themselves on the planet, and then we start to have the eukaryotes. And we'll talk about how the evolution from a prokaryote to a eukaryote took place. So prokaryotes tend to be smaller than eukaryotic cells. They are going to lack internal structures surrounded by membranes. So I said that word organelles before. Organelles are going to be these little organs or subunits within the cell, but they are membrane bound. Okay, and so prokaryotes do not have those organelles inside of them. Okay, they basically are going to have all the same abilities as a eukaryotic cell, but nothing is going to be separate from each other. It's all going to be kind of within the main chamber of the cell. So they lack those organelles, and they're also going to lack a nucleus. So the DNA is not going to be protected inside the nucleus like it is for eukaryotes. And they're all going to have a cell wall that is rigid. So the cell wall is going to be on the outside of the plasma membrane and is going to provide protection for these prokaryotes. So here's an image of kind of a stereotypical prokaryotic cell. You're going to have the kind of capsule on the outside that is going to have a cell wall that's going to provide kind of this rigid structure and protection. It's going to have DNA that is kind of held together in something called a nucleoid, but is not surrounded by a membrane. It's going to have little ribosomes, and it's going to have the cytoplasm. Most prokaryotes are going to have some type of propulsive element. So these are called flagellums that look very similar to what sperm have, and these allow it to swim so that these are able to move from different locations. Okay, and so you can read kind of a little bit more about prokaryotic cells and see how they're different from eukaryotic cells. So now let's talk about the eukaryotes. So eukaryotes are going to have organelles, and that's why when you look at them, they're all complex. They have all these different structures inside of them, and they're going to provide certain functions. Okay? We will name the different organelles and talk about what they do. Okay? One of the most important organelles is going to be the nucleus, and the nucleus is going to house the DNA. So right here, this red structure is going to be the nucleus. It is actually a double membraned structure, and the DNA is going to be stored in here. You can't really see the DNA, they're not showing it, but they do show this other structure called the nucleus, which I will mention what the nucleus is. So eukaryotic cells are very similar in that they're going to have all the same kind of organelles, they're going to have a nucleus, and this is going to kind of allow them to perform all the different functions that they're going to have to perform. Now, the region between the nucleus and the plasma membrane is going to be filled with a fluid, which we again call cytoplasm. Cyto meaning cell, plasm, think of like plasma, which is what your blood cells are in. That is going to be the fluid that is inside of the cell. Cytoplasm is going to consist of various organelles. So the organelles are going to be suspended within that fluid. They are not really floating around. They're more structured because we're going to talk about something called the cytoskeleton, but they are going to be kind of suspended within the fluid of the cytoplasm. Now, we really need to talk about the membrane because the membrane that is around the cell is going to be extremely important. We talked a little bit about phospholipids on the previous lecture, but we are now going to talk about what we call the plasma membrane, which is going to kind of make a barrier between the cell and the external environment. So this is going to prevent kind of things that are not supposed to go inside of the cell to enter, and it's going to allow some things to pass through. And so we'll talk about how that happens. But this is protecting kind of the living cell from the non-living surroundings. Now, within the plasma membrane, we have a very complex structure. There are proteins that are going to be there. We have lipids, we have phospholipids. They're all going to be kind of mixed in and create this very complex structure that is going to provide protection, but also allows specific communication with other cells and let some things move through the membrane and other things not. So the lipids are going to be very special, and they're going to be phospholipids. And we've talked about those phospholipids before. They have a phosphate head, and they're going to have that fatty acid tail. Now, the phospholipid forms this two-layer membrane called the phospholipid bilayer. And the reason why it does this is because the phospholipid has a very unique structure. So let's examine the structure of the phospholipid bilayer a little bit closer. Here we have what we call the phospholipid, and the phospholipid again is going to have this phosphate on top, and it's going to have these tails that are fatty acid chains. Now, the very interesting thing about phospholipids is that the head is going to be hydrophilic, which means that it interacts with water, and the tail is hydrophobic. So the reason why it forms a lipid bilayer, or two layers of this, is because the tails do not want to come into contact with water. So instead of having one side that faces water and this side facing water, because the inside of the cell is also going to have a lot of water, the tails point towards each other, and we have this bilayer. So you have two layers. You have the phospho heads, you have the fatty acid tails, you have the fatty acid tails, and the phospho heads. So it arranges like this in order to avoid being exposed to water. So most membranes are going to have specific proteins that are going to be embedded within the phospholipid bilayer. These proteins can help regulate traffic across the membrane, but they also have other functions such as kind of cell identification and cell communication. Now, if we look at this lipid bilayer, again, you can see kind of the red phosphoheads and the yellow tails, but these blue structures are going to be what we call proteins. And so the proteins are going to be embedded within the lipid bilayer. Sometimes they will span the entire length of the lipid bilayer. Other times they will just be embedded on one side, and some of them will only touch. They'll be called peripheral proteins, where they don't actually go in within the membrane, but are just on the edge. Now, I want to just bring this up, but the reason why they're arranged in this way is because of the chemical structure of these different proteins. So parts that are embedded within the lipid bilayer are going to be hydrophobic because they don't want to interact with water, and they're going to be in this area with other things that don't want to interact with water. The heads that are exposed to the outside of the cell and the inside of the cell, which are largely going to have water, those are going to be hydrophilic because they are able to withstand being around water. Now we have some of these things called glycoproteins, which are going to be carbohydrates that are attached to proteins. They often are going to be important for tagging cells for identification. The plasma membrane is often called a fluid mosaic because it is going to be fluid in that the different molecules can move freely past one another, and a mosaic because there's a diversity of different proteins and phospholipids that are going to kind of make up this membrane. So all cells are going to have a plasma membrane, but some types of cells are going to have an additional layer that is going to be called a cell wall. So plants, for example, have a rigid cell wall that surrounds that plasma membrane. Now that plasma membrane is fluid and does let water in. So if you have something like a plant that can't control taking water into its cells, it needs to have some protective structure to prevent the cells from popping if they get too much water. And so that cell wall, which is made out of cellulose, is going to help prevent those cells from kind of popping and losing their shape. So the made of that cellulose, that's the thing that we cannot break down in our body, so we can eat different plants parts that are made out of cellulose, but we just don't actually digest it. Okay? It protects the cells and it helps maintain their shape as well. And mainly protects so that they do not absorb too much water. Animal cells are not going to have cell walls. So this is a big, important concept. When we start talking about the different cells and I say something like, well, what are differences between animal cells and plant cells? One of the very first things that should come to your mind is that animal cells do not have cell walls. Animals do not make cellulose. We are going to only make glycogen, which is a storage carbohydrate, but cellulose is not something that humans or any other animal can produce. Okay? Instead, they are going to have an extracellular matrix, which is going to help kind of keep the cell together and the tissues organized. Okay, this will help protect them, and we will start talking about what the extracellular matrix is going to be made out of. Okay, when you see this image right here, what we're trying to show is that some cells are actually going to have connections between them, and so these can be called cell junctions. So they connect one cell to another. They can communicate through these cells and provide structure. We're now going to move to the nucleus and ribosomes. So these are going to be two structures that are really important in the genetic control of the cell and also producing the proteins. So the nucleus is going to be, you can think of it as the chief executive or the brain or the control center of the cell. It is going to be where all of the directions are going to be held, and it's very important that those directions are protected from outside entities. Okay, the genes that are located on the DNA are necessary for making the proteins, and so you want those directions to be protected so that you will always have a copy of the directions for those proteins. Proteins are going to do all of the work for the cell. They are going to help chemical reactions. They're going to actually be structural. There's basically nothing that a protein doesn't do other than carry the actual genetic material. So let's look a little closer at the structure and function of the nucleus. The nucleus is going to have a double membrane. So what you're going to see is if we look at the nucleus, which is this purple structure right here, there is a double membrane. And so this is cut away and you can see that there are two sides of this membrane. We call this the nuclear envelope, and it's a double membrane to help protect the DNA that's inside. So the DNA is kind of harder to see in this, but you're going to see some of this darker purple, and that would be the DNA. Now, there are pores on the outside of the nuclear envelope, and this is going to allow materials to pass into and out of the nucleus. Now, there are certain things that can enter and leave the nucleus, and others that are not going to be able to leave. One of the things that is not supposed to ever leave the nucleus is going to be the DNA. So the DNA stays in the nucleus, but it has all of the instructions for the proteins. And we're going to talk about the ribosomes in a second. The ribosomes, which are going to be important for making the proteins, they are outside of the nucleus. And so we'll talk about how we produce something called messenger RNA that is a copy of the DNA directions that can actually leave through these nuclear pores and go out into the cytoplasm to get to a ribosome. Now, inside of the nucleus, we have another structure called the nucleus. And the nucleus is this specialized, and it's very dark on the inside. So if you see a cell under a microscope and you can see the nucleus, and then you see an even darker spot within the nucleus, that is the nucleus. That is a specialized region where ribosomes are made. I wanted to mention a little bit about the DNA that's stored inside the nucleus. So the nucleus is going to have all the DNA molecules, but the DNA is going to be arranged in what we call chromatin. And when it's in its chromatin form, it's not just DNA, it's going to be DNA that is actually wrapped around proteins. And we call this chromatin. So chromatin is the DNA plus these proteins, and these are actually called histones. They're proteins that allow for the DNA to be wrapped, and then they form this kind of highly organized structure called a chromatin, and those can then be even further wrapped to form something called a chromosome. So we will talk a lot about chromosomes when we get to kind of some of the future lectures, but for now, all you need to know is that the DNA is going to be stored within the nucleus in kind of the chromosomal form. Now, the number of chromosomes in the cell will depend largely on the species. So for example, humans are going to have 23 pairs of chromosomes, but other animals and other plants are going to have different numbers. So let's now talk about the ribosomes. So ribosomes are going to be these special enzymes that are going to take the instructions from the DNA and turn them into proteins. Okay, so they're going to be responsible for protein synthesis. So that is their function. They're going to be made in the nucleus, but they are then going to be shipped outside of the nucleus and assembled in the cytoplasm. And there's actually two different components of the ribosome. They have a large subunit and a small subunit, and they come together inside of the cytoplasm. So the ribosomes will assemble the proteins, and we will go in depth about how protein synthesis takes place. But the ribosomes can either be suspended in the fluid of the cytoplasm, or they're actually going to be stuck in place on another structure called the endoplasmic reticulum, or you can call it the ER. That's another organelle that we're going to talk about. Now, here you can see an electromicrograph image of the cell. And what I want to point out are that these kind of orangish brown structures are going to be the ribosomes. And the ones that kind of follow the line, these are the ones that are going to be on the surface of that endoplasmic reticulum. But you also will have ribosomes that are just freely floating throughout the cytoplasm. Okay, and so you have two different types of ribosomes, ones that are stuck to the ER and ones that are floating. And they actually have different functions. They'll make different types of proteins. So here is kind of a very brief overview about how we go from DNA to protein. We can see the DNA is in the cell within the nucleus, and we have something that we make called RNA. The RNA can then leave through that nuclear envelope pore, and that can then go to the ribosome where we can make some protein. And remember, proteins are going to be made out of monomers of amino acids. Moving on from the nucleus, we are now going to go into the endomembrane system. And so all of these organelles are going to have membranes around them, and they basically are a manufacturing and shipping department of the cell. So these structures are going to be where proteins are made and other materials, and they're also going to ship those different materials to either parts of the cell or actually to other cells or other parts of the body. Now the endomembrane is going to be interconnected, and actually the first structure that we'll talk about, which is the endoplasmic reticulum, is directly connected to the nuclear pores. And so if you look up in this top, this is the nucleus right here. You can see it's double membrane. Here is a pore, and the pore is actually going right directly into this structure, which we call the rough endoplasmic reticulum. We'll also talk about the smooth endoplasmic reticulum and something called the Golgi body. Now, you can move materials directly through the actual membranes, or they can be transferred through different segments between them. And they're going to transfer through things called transport vesicles. So let's start with the endoplasmic reticulum. So there are two major parts of the endoplasmic reticulum. Okay? This is going to be where a lot of proteins and a lot of other materials for the cells are going to be made. So I want to point out again, here is the nucleus. We have nuclear pores, we have the nuclear envelope. And kind of continuing from the nucleus is this first structure, which we're going to call the rough ER. The reason why it's called the rough ER is because it's studded with those ribosomes. So it has these ribosomes stuck on the surface, and that is going to give it kind of this rough looking appearance. We then have the second endoplasmic reticulum area, but this one is going to be called the smooth endoplasmic reticulum because it lacks those ribosomes on the surface. Okay? The ER is going to produce an enormous variety of different molecules, and again, it is going to be composed of two parts, the rough and the smooth. So this image shows how the rough endoplasmic reticulum sort of looks like these sheets kind of folded on top of each other, or these little membranes kind of overlapping and folded. And so here is a picture from a transmission electron microscope, and you can actually see this structure. However, recently they've discovered that this is probably not exactly what the endoplasmic reticulum looks like, the rough endoplasmic reticulum specifically. So they've done new imaging that's shown that the endoplasmic reticulum is much more complex than it was previously imagined. And so what we're seeing here is kind of all the different tangled webs of the endoplasmic reticulum. So instead of it being these flat sheets piled on top of each other, it is a very complex structure. And this again is because we have new technology that allows us to kind of image and visualize what these different structures look like on a very small level. Now, most textbooks are going to still have this kind of stereotypical appearance of the endoplasmic reticulum. It's not too important, but I just wanted to mention that it does look different than we previously thought. Now again, the reason why it's called the rough ER is because there are ribosomes that are studded around the outside of the endoplasmic membrane. Okay, those ribosomes are going to produce membrane proteins and secretory proteins. So the rough ER is going to produce these two types of proteins, so proteins that are actually going to be kind of secreted outside of the body, and it is also going to produce membrane proteins, so proteins that are going to be embedded within the plasma membrane. After materials are produced in the endoplasmic reticulum, the rough endoplasmic reticulum, they're going to be packaged in these kind of spherical, they kind of look like little balls of transport vesicles. And so they're going to be a little ball that has a membrane on the outside, and on the inside is going to be the protein. It's going to move the protein. I think of transport vesicles as shipping boxes. So when you get a package from kind of UPS, it comes in that box, and inside is the materials, and so the transport vesicle is that box. So here we have kind of how this is all working. The ribosome is going to be right here. The ribosome is going to be able to translate and make this protein. The protein is made, and then it's going to be transported in this vesicle. And the vesicle is like a little box, or in this case, a little ball that is going to transmit this protein to its desired location, which it turns out this one is a secretory protein, so this is going to leave the cell. The smooth ER is going to be quite different from the rough ER. The smooth ER doesn't have any of those ribosomes on the outside, and because of that, it does not produce proteins, because ribosomes make proteins. And so if you have a smooth endoplasmic reticulum, there's no proteins going to be made without those ribosomes. Now what happens in the smooth endoplasmic reticulum is this is the location for the production of lipids, including steroids. So the steroids what we were talking about in the previous lecture, many of them would be produced in the smooth ER of the cells. These cells this organelle will also help in the liver detoxify circulating drugs. So when you take medication or other types of drugs, you don't want it to be continuously circulating within your body, and so the smooth ER will be one of the locations where those drugs will be kind of put into the smooth ER they'll be broken down and modified chemically so that they're no longer toxic, and usually they are tagged for removal so that they get out of the body. Now, the next structure that we're going to talk about is the Golgi apparatus, which is kind of a continuation, or it has a partnership with the endoplasmic reticulum. So the endoplasmic reticulum is making these different products, but the Golgi apparatus is going to be a shipping department. So it's going to take in these different transport vesicles. It's sometimes going to refine and actually modify those proteins or other materials that are being made, and then it will ship it to other parts of the cell or even to the plasma membrane so that it can be released from the cell and go somewhere else in the body. So this is what the Golgi apparatus looks like. It is kind of a series of these membranes. They're kind of stacked on top of each other, and they form vesicles. So basically what's happening is that all of this is made out of that lipid bilayer, so it has 2 layers, and the lipid bilayer will kind of just surround the different products and bud off and go to different locations. So now we're going to transition to some different organelles. The first is going to be a lysosome, and a lysosome is going to be a sac that is filled with digestive enzymes that are going to break down different molecules and debris so that it can be recycled or removed. Okay, these enzymes will break down large molecules such as proteins, polysaccharides, fats, and nucleic acids. So when there are things in the cell that are no longer functional and we want to get rid of it, the lysosomes will actually engulf those different molecules and break them down into their monomers in many cases. So here is an image of what a lysosome would look like. Lysosomes are going to have a bunch of different molecules inside of them that are going to be broken down. So for example, this kind of looks like a bacterial cell. Some of this could be a protein. These are going to be broken down so that we can recycle and reuse the different monomers. So here we have a lysosome, and it is going to have those digestive enzymes. So enzymes, again, are proteins. They're going to, in this case, break down different materials. For example, this is a food vacuole. So if we're eating food, the food is broken down in our stomach, and then the different parts can go off into the cells, and we can have the fusion of a lysosome with a food vacuole to start to break down and digest that food material. We also will use lysosomes to break down damaged organelles. So for example, this is the mitochondria, which we'll talk about in a little bit. This is an organelle that is no longer functional, and so it's going to be broken down so that its parts can be used again. Here's an actual image of a lysosome that has an organelle fragment, and this also looks like an organelle fragment. So it's breaking these down so that it can recycle its parts. Now, the lysosomes will not only recycle parts of the organelle or the cells, but they also will attack harmful bacteria and things that are not part of our bodies. So anything that's unwanted, both damaged organelles or harmful bacteria, can be broken down and destroyed. Vacuoles are another structure that are membranous sacs, but these ones are not going to be filled with those digestive enzymes. So we can have different types of vacuoles, but they can bud off of, for example, the endoplasmic reticulum, the Golgi body, and they can even bud off from the plasma membrane. So they sometimes will have different materials inside. This is a vacuole in a plant, and this vacuole will hold excess water and sometimes poisons. Here are other vacuoles in this cell, and they can kind of hold other materials in them. So vacuoles can also be used for locomotion. So there's something called a contractile vacuole that some protists, which are eukaryotic single-celled organisms, they can access or kind of pump out water, and that will help them move. Central vacuoles can also, again, in plants, store nutrients, absorb water, and sometimes will contain pigments or poisons. Here is a protist called a paramecium, and you can see the vacuole right here that is going to contract and release water out. So to review, these are all the endomembrane systems. They're all interconnected and they all have that membrane around them. So we have the nuclear envelope that's going to surround the nucleus. We're going to have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. We're going to have transport vesicles. We're going to have the Golgi body, or the Golgi complex it's sometimes called. Lots of different lysosomes and other vesicles all in here. And all of them are going to be made out of kind of that membrane material. We're going to move on now to two specialized organelles that are going to be energy converters. So we're going to have one that's found in both plants and animals, and one that's found only in plants. And we'll talk about kind of which one is which. So cells are going to require a constant energy supply in order to perform life. If the energy supply is cut off and stops, that cell will die. So we have two organelles. We're going to have the mitochondria, which is found in both plants and animals, and we're going to have chloroplasts, which are only found in plants. Now, chloroplasts are always depicted as green because they are actually green in real life. Some of these are just colorized, like mitochondria are not actually purple. But chloroplasts are actually green because they're going to be important. The pigment is going to be able to absorb sunlight, and we'll talk about that when we go over photosynthesis. So chloroplasts are going to be extremely important, and most of the living world is going to run off the energy that is provided by the process of photosynthesis. Okay, photosynthesis is going to be this energy process where we convert light energy into chemical energy in the form of glucose. So plants will make this glucose, and they make it from sunlight, and we'll go all over that entire process. And that sugar then can be eaten by us and other animals, and we can use it for our energy. So chloroplasts are the organelles, and they are these green little circles. These are organelles that are going to perform photosynthesis and do that conversion of light energy into chemical energy. The chloroplasts are going to have three major compartments within them. They're going to have kind of these double membranes that you can see. It's going to have this inner fluid, which is called the stroma, and it's going to have a different type of composition than we would have in the cytoplasm. We also are going to have the structures called granum, which is they're kind of these flat stacks, and individual members of that stack are going to be called thylakoid. Okay, we will go over all about this structure and how it functions in the future, but all you need to know for now is that chloroplasts are going to be involved in photosynthesis. Here is a cartoon and also a real image of a chloroplast, and you can kind of see the structure of the granum right here. So these structures are similar to what we see here. Mitochondria are going to be another energy converter, and these are going to be found in both plants and animals. They're the only things that we have. So we only have mitochondria. We do not have chloroplasts. So mitochondria are going to be the site of cellular respiration, which is going to take glucose that's made from photosynthesis and break it down into a new molecule called ATP. And ATP is the only energy source that we can use directly to power our cells. Now, mitochondria are going to be found in most eukaryotic cells, so that's why you can find it in plants and animals. And it's going to have kind of an interesting structure where it has a double membrane, and it's going to have kind of these different invaginations where you have an inner membrane and a fluid within. Okay, and the fluid is going to have a very specific concentration of different molecules. We have these kind of invaginations, which are going to be called cisternae, and the inside is going to be called the matrix. Okay, the envelope of the two membranes encloses the mitochondrion, okay, and they consist of a smooth outer membrane, an inner membrane that has those infoldings. Okay. One thing I love to talk about when we start to mention the mitochondria and the chloroplasts is the fact that chloroplasts and mitochondria are the only other structures in the cell that have DNA. Okay, so every other organelle will not have its own DNA, but mitochondria and chloroplasts have their own, which tells us something about their origins. So it turns out that both mitochondria and chloroplasts were originally single-celled organisms that were on their own. Okay, so they're free-living and they were prokaryotes in the distant past. However, what we think happens is that we had larger cells that then engulfed either the mitochondria or the chloroplasts. They engulfed those, brought them into their bodies, inside of their cells, and kept them and didn't digest them. And so what they did is they kind of took these energy sources, put them in within their cell, and were able to use them kind of for that production of energy. Now, this is an image of mitochondrial DNA. Mitochondrial DNA is quite a small amount. It's only 6,000 nucleotides, which sounds like a lot, but it's actually not very many if you think about it. The other interesting thing about mitochondrial DNA is that you only have maternal mitochondrial DNA. When you have sperm that fertilizes an egg, the sperm does not transfer its mitochondria to the egg. It only transfers its DNA. And so the mitochondria that you have is the same mitochondria you get from your mother. And you can actually trace back ancestry through mitochondrial DNA. Here is the chloroplast DNA. This is specifically from Nicotiana tabacium, which I think is nicotine. And it is going to have a more substantial number of bases. So this one is actually in 155,000 base pairs. And this again is showing that at one point the chloroplast was probably living on its own and was then engulfed within another cell. So this is what we call the endosymbiotic relationship of the mitochondria, and we think this also happened with the chloroplasts. So you had a primitive eukaryote, so this is a very early eukaryote that did have a nucleus, and we have this thing called an autotrophic prokaryote. Okay, and so this would be something like a mitochondria, has all the functions of a mitochondria, but it's living by itself. The eukaryote then engulfed that prokaryote, but didn't break it down and digest it. Instead, it kept it inside of its body, and it then started to use the function of the mitochondria to produce energy for itself. So it kind of became a symbiont, which means that it no longer can be free living. It now has to live within that larger cell. We're now going to transition over to the cytoskeleton. And so again, I've mentioned this multiple times, but cyto means cell, and this is a skeleton, so this is going to be the skeleton of the cell. Now this is going to be very different from what you would think of a normal skeleton, which is made out of kind of calcium and our cells. This is going to be made out of fibers that are going to extend throughout the cytoplasm. And so both of these images are showing you kind of all these different, looks like branches, all interweaving, and these are going to be the cytoplasm fibers. So the cytoskeleton is going to provide mechanical support to the cell. And so this allows for the cell to maintain its shape and kind of its placement in space. So it's not going to just be floating around. It's not going to lose its shape. It's going to, not collapse. We also can see that the cytoskeleton is going to be surrounding the different organelles and holding those organelles in place. So like I mentioned before, the organelles are not kind of like stuck in one spot, but they are going to be surrounded by cytoskeleton, which is going to keep them in relatively similar locations that they're always in. So they're not going to be able to move very far. They're going to have to kind of stay within a certain area. Now the cytoskeleton is going to have multiple different fibers, and these are going to be proteins. Okay, they are going to have different names. We can call them, some of them are going to be called microtubules that are going to have straight and hollow fibers. They are often going to guide the movement of organelles and chromosomes. So if we're moving organelle from one place to another, this will kind of help act as a guide wire for those things to move. Another fiber is called intermediate filaments, and there's another one called a microfilament. These are thinner and more solid, so they're not hollow like the microtubules. So here we have different images showing the different types of tubules and filaments. We have actin filament. This is actually very important in muscle contractions, but it also is going to be kind of important in how the cells are structured, so all cells. This is going to be intermediate filament, and you can see how they have different, this looks kind of like a piece of licorice to me, and this is going to be a microtubule, and so this is going to actually be hollow on the inside. And so all of these are going to help provide different structural support for these cells. Now I love showing this picture because this is a microtubule, and it's got a motor protein on here that is attached to a transport vesicle. And what's happening here is that this little motor protein is actually going to be able to walk across this tubule and kind of transport that vesicle that has maybe proteins in it or other molecules from one place to another. And it's just really interesting. There are videos of this that you can find on the internet. But it's going to move things from one place to another. So those vesicles that we just talked about, which had proteins in them, for example, they're not just floating around and happening to bounce into the plasma membrane to leave it. They're actually being transported by different proteins within the cell. Here are some examples of how kind of cytoskeleton and the microtubules will function in other types of organisms. So here we have, this is called a amoeba. An amoeba is a type of organism. It's a single-celled organism. It is a eukaryote because it has a nucleus, and it's going to have all these different microtubules in its cytoskeleton that allow it to move around and basically kind of send out these shoots so that it can move its body from one place to another, kind of building out its microtubules in one direction and then kind of moving its body very much like I feel like there's a scene in Monsters, Inc., the first one, where there's like this, I don't know, what is it called? Slime guy that's kind of moving in the same way, where it kind of extends part of its slime and the rest of the body follows. So here is what that looks like. So both of these are amoebas, and they are moving through what we call cytoplasmic streaming. But the way that they are building these structures, which we're going to call pseudopods, is by sending those microtubules kind of out into those different regions, and it can move its whole body that way. Now, two other structures that are within the cytoskeletal category are going to be cilia and flagella, and both of these are going to aid in movement. Flagella are going to propel cells in a whip-like motion. So whenever you think of flagella, I want you to think of a sperm. So a sperm is going to have a head and then a tail, and the tail is the flagella. Cilia are going to be these very short hair-like structures that beat in a coordinated fashion. And so you can see them in this lower GIF down here. So these are going to be found in different parts of the body in humans. So we have them in our upper respiratory tract, so that all of the mucus that is in our respiratory tract goes up towards our mouth so that we can swallow it and not down into our lungs. We also are going to have cilia that are present in the fallopian tubes and in the, they're going to help actually move the egg from the ovary into the uterus. Okay. And both of them have very similar architecture. They're just kind of different sizes and a little bit different shape. Here is the human trachea, and these are some of the cilia that are going to help sweep the mucus out of the lungs, and these are some epithelial cilia. I think specifically this is in the uterus. Okay, and here are some sperm. You can see their tails that allow them to swim, and so these are flagellums. And here we have a combination. We have the flagellum and the cilia. So in this situation, we have, we're inside of the vaginal canal. We may actually be in a fallopian tube at this point. We have the cilia that are part of the fallopian tube that are going to help move, in this case, not only the egg, but also the sperm towards each other. Now, this is a really cool video if you want to take a look at it. The link will be on the slides that I post, so I don't think you can press this if you're in the video, but it will show all different aspects of the cell in kind of this cool video. The last couple slides are all going to be showing different parts of the cell so that you can identify them if you would like to print these out and put them on note cards. So for example, this is going to be the plasma membrane. That is the nuclear membrane and the nucleus. Remember, inside in the very center is going to be the nucleus. We have ribosomes that are both on the rough endoplasmic reticulum, but also free floating. We have, now this is highlighting the endoplasmic reticulum and both smooth and rough. We have the Golgi body. These are different types of vesicles or lysosomes. Here is a vacuole for plants, and you can see that this is a plant cell with its cell rigid wall. Here are chloroplasts, and here are some mitochondria. These are going to be part of the cytoskeleton. This is a flagellum. And here is a table comparing the two groups that we talked about earlier, the prokaryotes and the eukaryotes. Okay, and so that is going to be the end of this lecture. Make sure that you are able to compare prokaryotes from eukaryotes. So what do they both have? What is different about them? Be able to also compare animal cells with plant cells. So what is something that's special for plant cells? Well, they have a cell wall and they also are going to have chloroplasts. So be able to make those comparisons.